Okay, this is video number 43 of the Diaries of a Coronavirologist YouTube channel. Today is still the 25th of November, and we're still at 60 million confirmed cases of COVID-19 around the world and around 1.4 million confirmed deaths. Now, the reason I'm saying still there is because this is my second recording of a video in the same day, but I'm posting them on two separate days onto YouTube. Because in my last one, it was quite long, and I was taking apart the data for the Oxford University and AstraZeneca vaccine and talking about all the issues that there are with that vaccine data and the fact that they're applying for regulatory approval with said data. So there are issues that I was highlighting there. There are also broader issues and broader concerns about the vaccine development process that's going on at the minute. So I've had people directly ask me, and I've seen things on social media asking good questions about this general process. So in this video, I want to talk about some of those ideas. So I've broken that down into four main topic areas and then one final little piece of some other questions and things we'd love to know that I'll just run through very quickly. I've timestamped all of those below, so you can jump around if you want different topics, or you can come back to them easily enough should you want the information on specific areas in future. So the ideas I'm going to talk about are broken down into side effects, so short-term and potential for long-term side effects that we're unaware of, the issues about whether this vaccine is being rushed or not, and therefore whether we can trust what's coming out. There's this idea going around that it's too rushed and therefore we shouldn't be taking it. There's an idea about how long-lived the immunity is, and therefore will we need yearly vaccination like we do for influenza. And then I'll do a little discussion about the difference between DNA and RNA-based vaccines. So again, if you watch my last video and the ones before, and you've been following anything about vaccines recently in the news, you'll know that the Pfizer, BioNTech, and Moderna vaccines are RNA-based, and the Oxford University AstraZeneca and Sputnik 5 vaccines are DNA-based. So I want to talk a little bit about the differences there. And as I say, the final piece is just and some other things we'd love to learn about as time moves forward. Okay, so I'll start with side effects as the first topic for the discussion in this video. So there are some side effects from vaccines, but these are largely very mild, and that's what we're seeing here with these vaccines. So if we look at, say, Pfizer, BioNTech, and Moderna, there's reports of some mild symptoms that come from the vaccine. So these are things like developing a mild fever for a few days, developing a headache, developing some fatigue or muscle soreness, especially at the site of injection. But most of those are occurring in under 5% of people. So they're a very limited number of cases, but they are some discomforting things. But the concern there is the fact that those kind of symptoms resemble COVID-19. They resemble a mild case of COVID-19. So a concern, and something I want to convey, is that just because you might get this vaccine year down the line, whenever it is, and you develop those symptoms, it doesn't mean the vaccine isn't working. That's just your body mounting the immune response that is going to protect you from COVID-19. That's a normal reaction, especially something like a pain at the site of injection. Now, why this is a bit of a concern if people start to think the vaccine is not working, start to think they've developed COVID-19 after one injection, is the fact that all of these vaccines so far require two injections. Again, if you watch my previous video, you'll know this idea about prime boost vaccination. So you get a priming injection, you wait about a month, then you get your booster injection. Now, all the numbers talking about efficacy for these vaccines, so Pfizer, BioNTech at 95%, Moderna at 90%, that's after 2 doses. There's no information about the protection that comes from just 1 dose alone. So if people start to think that the vaccine hasn't worked and they've been infected with COVID-19 because they developed a fever and a headache and those kind of symptoms and don't go and get their second vaccine, that's going to be a big issue for all of these vaccines. So it's something that needs to be conveyed, something I want to convey here, that there are these mild symptoms that do seem like COVID-19, but they are not. That is just the vaccine working. Now, the other part on side effects is the idea of more long-term side effects. So those are short-term side effects. When you get the injection, you develop a fever over the next couple of days or whatever it may be. But there's also questions I've seen floating around about how do we know there aren't long-term side effects from these vaccines? These trials have only been going for a couple of months, and now we're about to try and put them into millions of people and potentially eventually billions of people if we're going to really get high coverage with these vaccines. Now, the thing is, there aren't really well-established examples of long-term side effects from vaccines. Vaccines, typically, if they're going to have side effects, they are acute side effects. They're the things that appear within the first week or two after vaccination. And if they are severe, then that vaccine never makes it all the way through to regulatory approval, be that emergency usage agreements or further. So there are very few examples where long-term side effects appear. And also, while these phase 3 trials have only been going on since the summer and are now reporting data here in November, there's also been the phase 1 and phase 2 build-up. So these vaccines have been going into people since springtime, and those people are still being assessed. So we do have an idea about 6 months or so of vaccine data from people. Obviously not as many as are in the phase 3 trial, but there's no evidence of any development of any kind of long-term event in people that received the very, very first doses of these vaccines. Now, it would be wrong of me to ignore the fact that there have been examples of long-term effects. However, as I say, they're incredibly rare compared to how many vaccine doses are given around the world every year. An example I will bring up, though, is the Pandemrix vaccine. So this was a vaccine produced for targeting swine flu following the 2009 swine flu pandemic. It was given to people around the world, and the highest uptake of the vaccine was in parts of Scandinavia, and particularly in Sweden, where they achieved around 61% coverage. Now, it was found a few years after that vaccine started to be administered that there had actually been an increase in the amount of cases of narcolepsy, which seemed to track with the vaccine. So narcolepsy is a sleep disorder where you don't go into proper REM sleep, and therefore you mess up a lot of things because REM sleep is super important. And they found that in Sweden, the number of cases went from 1 per 100,000 people to 15 per 100,000 people in that time span following the injection of Pandemrix. Now, it's found that there was some association between the development of narcolepsy and Pandemrix, and it's possibly linked to the adjuvant which is used in the vaccine. So adjuvants are things that can help boost the immune response that you develop to the vaccine. And I will say none of the vaccines that are going around now, the RNA vaccines and DNA vaccines, have adjuvants as far as I've ascertained. It's all proprietary information, so it's hard to know at the minute, but I haven't seen any references to adjuvants in these vaccines. So in Sweden, the vaccine-associated cases of narcolepsy did seem to go up. It did seem to be driven by this vaccination, and it seemed to be more in children and adolescents and that age range. But the cases of narcolepsy were still only 1 in 18,400 people. So this is still a very, very low number. It is a long-term effect from that vaccine. It didn't appear straight away. It wasn't noticed straight away, but it's also very rare. So of course it took time for people to actually make this association. Now, I'm sure I'll get inundated with other examples, and if I follow it all, I'll go down lots of rabbit holes. But my point here is I do want to say long-term side effects from vaccines can happen, but are incredibly rare, and it's very hard to find good scientific data of these. And that's not because we don't look. When developing these vaccines, they continue to be assessed for years and years afterwards to make sure that they're working, to work out how long-term the protection is, and to see if there are safety issues that develop. So just because something gets approval doesn't mean it stops being assessed. So while there is a valid concern about the potential for long-term unknown consequences, it's a very rare and unlikely event that there will be. What does have long-term effects, though, is COVID-19. There are more and more examples and cases of people who develop this long COVID-19, these long haulers, who develop chronic fatigue symptoms. So these are people who have this mind fog, struggle to find words and things like that. There's people who have serious fatigue, struggle to get out of bed. There's also the people who develop issues in their circulatory system and damage in their lungs, which leaves them unable to breathe for a long period, properly for a period of time after their COVID-19 diagnosis is negative. So even if there are these really, really rare events of long-term issues from the vaccine, they're going to be nowhere near the level of the long-term effects that COVID-19 may cause. the likelihood of developing something long-term from a vaccine versus the actual virus just don't compare. So those two things combined, the fact that the long-term evidence for long-term effects from vaccination are very rare and very scant, and the fact that COVID-19 itself does cause long-term effects, and that's something we should be trying to protect against, really leads me towards the fact that long-term side effects from the vaccines that we don't anticipate, we don't see, very, very unlikely to happen. All right, my second topic for this broader issues around vaccine development is whether this vaccine's rushed. So I've seen things, I've been asked things about how have we been able to make a vaccine in under a year? Why have we been able to develop this? It must be rushed. How do we know it's safe? And all those kind of things. So while this has been the most rapidly developed vaccine or vaccines we've ever seen, that doesn't necessarily mean it's rushed per se. So we've known about coronaviruses since about the 1930s with the discovery of a coronavirus that infects chickens. And we've known about coronaviruses infecting humans since about the 1960s. Those viruses were just viruses that cause common colds, and still do just cause common colds. So there's no impetus there to produce a vaccine. There's no drive to spend that money because they just cause common colds, they don't kill people, they don't cause any severe disease. However, in 2002, 2003, SARS-1 spread around the world, and then in 2012, MERS emerged and had a relatively small outbreak around the world. And these viruses do kill people. So SARS killed about 10% of the people it infected, MERS has killed about 35% of the people it has infected. So with those, there came an impetus to start producing vaccines. With SARS-1, a lot of work went into trying to develop vaccines against that virus. However, it only spread for 8 months and then it disappeared. So vaccines were there just about ready to go to look to see if they're safe, look to see if they're effective, kind of what we're doing now. But the virus stopped spreading, so there was no way to work out if those vaccines were actually effective and able to protect people from SARS-1 infection because no one got infected with SARS-1 anymore. With respects to MERS, there was again a big push to try and produce a vaccine to this coronavirus. However, that virus itself doesn't spread very well between humans. It still occasionally causes cases, so therefore it's been spreading technically since about 2012, but we've only had around 2,500 cases. So while there was a push to produce a vaccine against MERS, there's not enough spread of the virus to again prove if it's actually protecting people because the virus doesn't spread well enough. So for both of those viruses, a lot of research went into producing a coronavirus vaccine. First 2002, 2003, that then got shelved and then got sort of reawakened in 2012, but could never be properly tested. So that then brings us into the modern day where we've got SARS-2. There's already been all this research that's gone into trying to produce vaccines against SARS-1 and MERS that can then be built upon and adjusted to SARS-2. For example, the Oxford University AstraZeneca vaccine I was talking about in my last video, that was originally set up to be a MERS vaccine. They originally were trying to vaccinate people against MERS. So the DNA in that viral vector was originally the MERS spike DNA that was then just replaced with the SARS-2 spike DNA, which is actually a relatively simple process to do in the lab. They'd already tested to see if that vaccine vector system was safe in people for MERS. It was a quick process to therefore go through and see if it was safe in people for SARS-2 and then to start the efficacy trials that we're now getting reports upon. And that isn't to say that steps are being skipped. These are still being assessed just as rigorously as any vaccine we've had in the past. The difference is we have this backbone or this bedrock of previous research on other coronaviruses from which to build upon quickly. To use that famous Newton quote, we're seeing further from the shoulders of giants, and that's why things have been produced so much more rapidly for this virus. Now also, I was talking about how we couldn't develop the vaccine to SARS-1 because it stopped spreading, and we haven't developed a vaccine for MERS because it doesn't spread very well. Well, this virus is pretty different, as is pretty clear. There's over 60 million people who've been infected with it. There's spiking cases all around the world. So it's quite easy to see if these vaccines are protective because so many people are being infected each day. So these vaccines have been produced rapidly, but not in a rushed manner because of the background knowledge we have. But also they've got never before seen financial backing and probably never before seen effort going into them. So there's a huge amount of money going into developing vaccines and developing treatments against coronaviruses. And the scientific field is uniting like never before to try and develop this. People have dropped their own research on lots of different viruses and are all focusing on SARS-2 now. So we have a huge amount of money, a huge amount of effort going into this. And the fact that we've got to the stage of developing vaccines within a year just goes to show what science can achieve with money and with directed effort. If science had a fraction of the money that goes into military budgets, notwithstanding that some of that does filter into science, we may not have even been in this situation of having SARS-2 as an outbreak because we may have had surveillance systems that detected it before, but I probably shouldn't go up onto that soapbox. My point is, this is rapid, but it's not rushed. We had a lot of background production, information, infrastructure from previous coronavirus outbreaks. There's a huge amount of money and effort and impetus to produce the vaccines. So it is rapid, but it hasn't skipped steps. It's gone through all the same rigorous safety testing in phase 1 and phase 2 clinical trials. It's done the preclinical research in animal models before it even went to humans. And now we're in a stage where we're looking at the efficacy data in phase 3 trials. And because the virus is still spreading so rampantly, we can develop knowledge quite quickly about whether these vaccines are indeed protective. And that brings me up to idea number 3. So this is an idea about how do we know how long-lived immunity is to this? Are we going to need a vaccine every year? Now, of course, at this stage, we don't know how long-lived immunity is from these vaccines. We don't even know how long-lived immunity is to natural infection from the virus, and that's been going on longer than we've been putting vaccines into people, because we just haven't had the time to work those things out. From other coronavirus infections, we can make an assessment that natural immunity from the real virus probably lasts about a year to 18 months, maybe a bit longer. But that kind of amount of time, and data so far seem to be showing that people generally immune out to about 6 months, possibly longer. With the vaccine, we may develop a slightly different type of immune response, and maybe it will be longer lived, but right now we just don't know. So in terms of whether we would need annual vaccination like we do for influenza, it's possible. However, if we get a good long-lived immune response, we maybe won't need it every year. Influenza mutates very rapidly, and it mutates to avoid the vaccine-developed immunity every year. Coronaviruses don't mutate anywhere near as rapidly as influenza viruses do, so potentially they wouldn't mutate rapidly enough to avoid the vaccine-induced immunity and therefore necessitate a new vaccine every year. But of course, that remains to be seen. We're only going to find that out after people start getting vaccinated and to see how long they're protected for. And again, this comes back to some more stuff about safety. Those people are going to continue to be assessed for that reason, to work out how often you need to give them the dose of the vaccine. So they're also going to be constantly monitored for safety and to work out if there are any long-term effects that start to develop, those super rare long-term effects I was talking about. We're going to find them. This research is going to continue for years and years down the line to work out how to dose these vaccines in order to protect people. And my final point of the video, or final main point, number 4 of the video, is the RNA versus DNA vaccine approaches. So Pfizer, BioNTech, and Moderna, those 2 vaccines are RNA-based, whereas the AstraZeneca Oxford University and Sputnik 5 vaccines are DNA-based. So generally, DNA is much more stable than RNA is, and that's why life on this planet is DNA. One of many reasons, I'm sure, but a main reason that life on the planet is DNA-based. So that means that DNA-based vaccines are a lot more stable and therefore are a lot easier to store and transport. The Oxford University vaccine can be kept in a fridge. I'm pretty sure Sputnik 5 is the same. They can be kept at 4 degrees Celsius in a typical fridge you have at home, and they're fine, and they can still be effective. Pfizer's vaccine, produced by BioNTech, that has to be kept at minus 70 Celsius, which is super cold, or minus 70 to 80. That's super cold compared to 4 degrees Celsius. Moderna's vaccine, not quite as bad. That can be kept at minus 20 Celsius, but again, that's your typical freezer you have at home. So DNA has the advantage of being much more stable. So that's an advantage of those vaccines that are being developed. Now, I also had someone specifically ask about, are there any concerns of whether the DNA vaccines could lead to DNA integration into the human genome, which could have untold side effects and mutagenesis and things like that. So this is a topic that would require quite a lot of explaining for the full-on background, but I do want to do justice because I got asked the question and I want to approach it. So to try and do it very briefly, if you imagine our DNA as a piece of string, I don't have one to hand, unfortunately. Imagine our DNA as a piece of string, and you take some scissors and cut that into pieces. It's possible to tie in another piece of string into that and then seal it all back together with a little knot, 2 little knots, and then it's just back to being normal DNA. So this is actually how HIV infects humans. HIV is an RNA virus. It makes its RNA into DNA. Then it has a virally produced protein enzyme, that chops our DNA up into 2 pieces, and then it sticks its DNA genetic material in there, seals it all back up, and tricks ourselves into thinking that that viral DNA genome is the human genome and treats it as such to then produce more viral particles. So this integration process of DNA can happen. With HIV, it's done by viral proteins. So when someone was asking me about could these DNA vaccines have untold consequences of mutagenesis from DNA insertion, that is something that can happen. However, there's very, very little evidence that adenovirus DNA can do that. Adenovirus DNA typically just sits next to our own DNA in the cell and is its own little entity. So because these vaccines are produced on an adenovirus vectored backbone, the genetic material that produces the vaccine protein, the spike protein, acts like adenovirus DNA. So the chances of integration are very slim. There are some low incidences where adenovirus DNA can integrate into other DNA, such as our human genome. But the thing is, the cells that get this vaccine are really likely to be killed. So firstly, they're going to be producing the spike protein that triggers our immune response, and that's a signal to kill those cells because the immune system thinks that they're infected with SARS-2, thinks, oh no, we've got to kill these, and then produces the immune response that's beneficial to us should the real SARS-2 virus come in. So they have one trigger that suggests they should be killed. But they also have the evidence that they were infected by the adenovirus. Again, this may make more sense if you've watched my previous video discussing this in a bit more detail, how these vector vaccines work. But there's evidence that the cell was infected by an adenovirus, even though no new adenoviruses are produced. And that's also a cue to the immune system that that cell should be killed. So it's incredibly rare that adenovirus DNA integrates, and therefore incredibly rare that DNA from these vaccines would go into our human genome and have this mutation potential. If it were to, there's also a really high chance those cells will be killed before there's any problems. So I don't think this is something to be concerned about with regards to DNA vaccines. DNA vaccines have the advantage that they're more stable than RNA vaccines. The only thing we really need to work out is which are more effective and which are going to provide protection and which can we distribute to enough people. Okay, and the final thing for the video is my sort of all clumped together open questions and things we'd love to learn about as time goes on. So I haven't been able to cover all the things. It would take me too long to cover all the different talking points. But just some examples, we need to work out things like how well this vaccine works in the elderly and those with underlying health conditions compared to people like myself who are younger and healthier, because the people with the underlying health conditions and the elderly are the ones who require the vaccine the most. So we need to make sure that it's really effective in those people because they're the ones who we really truly need to protect. We also need to work out things like whether these vaccines will drive mutation and evolution of the virus. So I was saying earlier that coronaviruses evolve and mutate not less rapidly than influenza viruses do, but that doesn't mean they don't evolve and change. So these vaccines are an evolutionary selection pressure which could drive evolution of these viruses, cause them to change to try and avoid our immune response. So we need to look at whether that's going on. We need to work out what viruses are appearing in people and whether they're mutating as a consequence of this, and therefore if we need to change our vaccine approaches. We also need to work out things such as whether or know about whether these vaccines that are going into people are preventing them from being infected outright, or whether they're just preventing them from developing severe disease. That's an important distinction, because if people are just being protected from developing severe disease, that's great for them, but they could still spread virus to people who aren't vaccinated. Of course, there's a lot of people who are going to choose not to take the vaccines. I'm not going to try and convince otherwise. I don't have the time and energy, to be honest. But there's also people who can't receive vaccines, people who are too young, people who have immunocompromised conditions and who it's not safe to give vaccines to. So if these vaccines are only protecting or only stopping the development of severe symptoms, but can still allow people to spread, that's something that has to be a consideration moving forward, because that won't allow us to stop the spread of the virus completely. It will just stop the severity of disease. So we need to learn things about whether people can still spread if they've got the vaccine, even if they're not developing symptoms. And it would be great to know why they're not 100% effective. What are the determining features of who gets protected and who doesn't? So Pfizer and Moderna are reporting around 90 to 95% protection from people who get the vaccine. So those people who actually get the vaccine and get infected, why is that? What are the determining features of the immune response that produce a protective response? That would be great to know. But like the other things I'm listing here, these are all things that are going to take time and will be the topic of future and ongoing research. And so those are the topics of discussion for this video. If there's other things, please, of course, drop them down in the comments and I'll try and tackle them either there or make another video like this in the future. But the bottom line is, at this stage, in under a year, there's been an amazing amount of work that has gone into producing vaccines. We've got the 2 RNA vaccines that are looking great, and we've got these 2 DNA vaccines that we need to know more about but are looking promising. There's other vaccines on the way that we'll hear about soon enough. Johnson & Johnson, who I keep forgetting about when I do my videos, have something on the way. A Chinese company whose name are escaping me right now, they've got another one on the way. We're going to hear about more and more vaccines hitting the endpoints of their phase 3 clinical trials. So we're in a really good position to be moving into 2021 with good news on the vaccine front. Of course, it's going to take time for the rollout to produce enough vaccines to get them into everyone who needs them, let alone everyone who may want them. So this isn't over. We still need to keep doing social distancing. We still need physical distancing. I want to keep stressing physical distancing. We still need to keep doing physical distancing, and we still need to keep wearing masks and doing all those things. But things are looking promising. So even though there are these broader concerns, hopefully I've helped to dispel some of them with some additional information. And I do really think we're in a positive position going towards the end of 2020. And that's where I'll end things, with much more recording from one night than I normally do, and the night before Thanksgiving. So thank you for watching. If you found the videos useful and interesting, please drop a like for the YouTube metrics. And if you're new around here, please subscribe, again, for those YouTube metrics. And feel free to share if there's people who you think would take value from these videos. I'm trying to put out good information, and the more people who get that, then hopefully the better. As always, please stay safe, wash your hands, wear a mask, and keep calm and carry on. We will get through this pandemic.